I'm Melinda Irwin. I'm a professor in the School of Public Health with Vasilis and also Deputy Director for the Cancer Center overseeing population sciences research, which focuses on lifestyle, genetic, and environmental risk factors for cancer etiology and outcomes. So we're delighted today to have the Yale and national international expert in environmental carcinogens and cancer speak to us, Dr. Vasilis Vasiliou, who is also, he's the Susan Dwight Bliss Professor of Environmental Health Sciences, as well as Chair of our Department of Environmental Health Sciences in the Yale School of Public Health. He received his PhD in biochemical pharmacology from the University of Ioannina in Greece. He then trained in gene environment interactions, molecular toxicology, and pharmacogenetics at the Department of Environmental Health and the College of Medicine at University of Cincinnati. He joined Yale almost 10 years ago in 2014 from the University of Colorado School of Pharmacy, where he rose through the ranks to become professor and director of the toxicology graduate program and was professor also of ophthalmology. He's established an internationally recognized research program that's been continuously funded by NIH since 1997, and his research interests include the etiology and molecular mechanisms of environmentally induced human disease, such as liver disease, obesity and diabetes, cancer, and neurodegenerative diseases. Vasilis is the director of the NIEHS-funded P42 Yale Superfund Research Center, and also the director of the NIAAA-funded R24 Resource Center for Mouse Models and Metabolomics Tools to investigate alcohol metabolism and tissue injury. Vasilis really does translational research from preclinical work to clinical to community-engaged research, focusing on environmental risk factors in cancer. Thank you.
Thank you very much, Melinda. Thank you for the invitation. And Melinda, thank you very much for the impressive introduction. I don't know if anyway, so I wish, let me start. Actually, the talk of today's lecture is Exploring Environmental Health, the Insights through our P42 Center, Research Center on Emerging Contaminants and their effects on cancer. So one of the concerns that we have is, you know, that 50% or maybe more than 50% of the cancers might not be due to the genetic effects, might not be to mutations, might not be to polymorphisms or things like that. So they have an environmental impact. One of the things that has triggered my attention the last 5 or 6, 7 years is the early onsets of cancer, which actually there was a very nice review in Nature Reviews in Clinical Oncology, which posed the question if the early onset cancer is an emerging global epidemic. And it has, as you can see, the incidence of cancers in various organs in ages less than 50 and actually less than 40 has been rising in many parts of the world since the 80s. The evidence suggests an etiological risk of risk factor exposures in early life, young adulthood, and although specific individual exposures remain to be largely unknown. So this is what my interests are, how these environmental exposures could lead not only to cancers, but also to early onsets of cancers. And we can go from liver cancer to colorectal cancer with associations to alcohol, which I could give you another lecture on that. But anyway, the early onset cancer epidemic might be one manifestation of increasing the trends in the development of many chronic disease in young and future generation. So what we come here is the early life exposome. It's the exposures of the mother, and actually now it's also exposure preconceptionally for both mother and father, what they have been into it and how this would affect the development of the embryo in the later studies. So the exposome includes environmental exposure, Melinda talked about diet, lifestyle, obesity, and microbiome. And this hexposome has changed completely in the last 40 or 50 years. So a lot of people looking into how these early life exposures could have an effect. Of course, we should not ignore the exposure we have on daily basis. And talking about that, one of the most important thing is drinking water. You drink your water, and in most of the cases, especially when you come to emerging contaminants, you have no idea what the water contains. So it's a lot of aspects in here. So talking about drinking water and protecting the environment. So the federal government has the Superfund Act, and what that is refers to a comprehensive environmental response compensation and liability act known as CLERCLA since the 80s. So what the federal government did is they put a law that provides a legal framework for cleanup sites of contaminated and hazardous substances. Especially, there are some states, Connecticut is a heavily industrial, actually it's a retired state of heavily industrialized area. If I'll show you the sites of Superfunds, either federal or local, we might walk away and go and look for a job in another state. It's everywhere. And I will explain you why, I'll give you some of these examples. Again, we have to be taking everything into consideration. So there is the federal law. If there is a company that has a spill out, so they're getting a fine, and that could be hundreds of millions of dollars. So this amount of money, some of this amount of money, they go to the EPA, and some of them, they go to National Institute of Environmental Health Sciences. And what the Environmental Health Sciences does, takes this money and creates the centers. So we have here the Cancer Center. At the same time, we have the Superfund research centers throughout the United States. And essentially, the centers focus on the understanding of health effects of the exposures to hazardous substances, developing innovative technologies to mitigate, essentially sites cleanup and engaging communities affected by these environmental issues. So their research contributes to the overall goal of safeguarding the public health and environmental in areas of hazardous waste contamination. So this is what are the centers, and this is, as I said, this is where the federal money from penalties on those cleanups go, and they come in. So how many centers there exist? We have 23 funded centers in the United States. And as you can see on the top left is our Connecticut, it's a Yale Superfund Research Center. This is the first ever center we got in Connecticut. And the focus of, as I said, the focus of our center is on emerging contaminants. So what are water contaminants of emerging concern are? These are chemicals that are detected in trace amount in our drinking water within global drinking supplies, that their risk on human health is not fully understood or even not been evaluated at all. Okay, and what are these emerging concerns include? They're including industrial chemicals, such as PFAS. Everybody has heard about the PFAS. Everybody, today, at least you will get educated on 1.4 dioxane, on some volatile solvents that they exist. But in addition to that, we have a lot of pharmaceuticals, personal care products, and actually yesterday, NIHS had a webinar about the exposome on personal care products. You'll be surprised how much they go down the drain and how much they end up in your drinking water. It's truly amazing. And of course, you have pesticides and herbicides that come from the agriculture that eventually, they're going to end up into the water table, and they're going to end up to our drinking water. So the overall objective of our Yale Superfund Research Center is to improve public health from the emerging water contaminants in the drinking waters. And what we're trying to do, we're trying to develop not innovative research in terms of the mechanisms of toxicities that these emerging contaminants cause, and also look at the ways that we can mitigate that, we can detect and mitigate that, and also inform the communities, talk with the communities, listen to their needs, and talk to us about it. Our center has been focused on 1.4 dioxane and also it's what we call, I'm sorry, I'm walking around. I'm Greek, I'm sorry. It's in my genes. This is in my genes, it's not environmental. So it's 1.4 dioxane and what we call its co-contaminants, which is it's the dichloroethane, trichloroethane, and also trichloroethylene. Those are volatile solvents that they exist, and I will explain you why and how this has come up with. But essentially, this is what our center is doing, and this is what we're focusing. Now you can say, why did you focus on that? So when I came here, it was 2014, I immediately went on, I think the next year, immediately, I met with the state authorities, the Department of Public Health and the Department of Energy and Environmental Protection. And I said, I'd like to develop a Superfund research program for the state of Connecticut. What is the issue that you have? At that time, PFAS were not that hot. And actually, thank God, because we took the direction of 1.4 dioxane. Had I known anything about 1.4 dioxane? No. But they told me that this was the major issue. And the major issue was because of these solvents on these multiple sites in the state of Connecticut. And the problem with 1.4 dioxane is they could not filter it. It would go through every aspect, and even in public water, they cannot filter it. If the source is contaminated, it will end up in your drinking water. So that was a major issue, and this is why we chose to go with 1.4 dioxane. So the Superfund Center, and actually I'm really proud, not because I got the grant, I'm really proud because I brought the 4 schools together, and that is our School of Public Health, the School of Medicine, the Engineering, and the Environment. And we developed this program. This program consists of, as you can see, 4 research projects and 4 cores. We have the administrative core, we have the training core, the DMAC, which plays it's the data management and analytics. We also have the community engagement, and of course, we have the training core that you will go. And I'll go in a little bit more details when we move forward on this. So again, one more time, it was the concern of the public institutions was on 1.4 dioxane for a particular reason, because it is a possible human carcinogen. It has been found that it causes cancer in animals, but now there is no epidemiological studies yet in humans. This is why it's emerging contaminant. And it's found with the other co-contaminants and has been prioritized by US Environmental Protection Agency on the third unregulated monitoring role for testing in 2013 and 2015. And you will see the map. It is spread throughout United States. So it's been also characterized as forever chemical, just like the PFAS, because it cannot be filtered. It's difficult to be, metabolized by bacterial species and so on, and it goes there. This is from the Chemical Environmental News saying that this is really. So when I put the team together, we wrote a big grant that we oversee what it was available, what was the scientific evidence, what was the epidemiological studies, what are the strategies to mitigate that. And we had that on a very nice review that actually helped us to get the team together. So 1.4 dioxane occurs in 3 isomers, 1.4, 1.2, and 1.3, but the concern and the most prevalent is the 1.4 dioxane. Is it a new chemical? No, it's not. I'm just not going to go in all details of this because we can talk about for a long time, but it was first synthesized in 1863. And initially, it was used as a stabilizer. It was used as a stabilizer for the solvents, the DCA, TCE, and DCA. And what happens is These solvents, they were covered on, they were transferred on aluminum containers. And the aluminum containers, you know, they have a cover inside, which is aluminum, and they protect from interacting with the metal. So what happened is, after a certain period of time, these solvents interact with the metal and they create even toxic products. So what they did is they found out 1.4 dioxane, it could block the catalysis of this reaction. So they were using 1.4 dioxane as a stabilizer of those chemicals, not only to protect the toxicity, but stabilize the solvents for their use. And what we're using this, anything that you can imagine, I'll show you. So it was used as a stabilizer to begin with, but later, as you can see from this, it has been used in many areas. So this is the uses. I'm not going to go in great details, but includes from the stabilizing to medical, pharmaceutical, rubber and plastic industry, printing inks and paints, adhesives, brake fluids, brake cleaning fluids, and also rust remover, and also antifreeze and de-icing. The stuff that they throwing on the airplanes before we take off on this, it has 1.4 dioxane, quite a bit. And what we end up with on the ground, okay? So pesticides, and some of the pesticides, they have up to 50% of 1.4 dioxane, 50%. Anyway, and also the consumer products we talked about before. What is environmental concerns? First of all, groundwater contamination. Resistance, it's, as I told you, it's there, it cannot be really degraded easily and can travel. It can travel everywhere. Toxicity, it's been classified, as I told you, as a possible human carcinogen by both the US EPA and the National Agency for Research on Cancer. Regulatory concerns, there are plenty of concerns. There is no federal regulation. Well, we don't have federal regulation yet, even for PFAS. And I don't know if you watched the movie Dark Waters, which I recommend that you do. You will realize what I'm talking about PFAS and regulatory issues. So as I told you before, where you find 1.4 dioxane, you find all the other solvents, or vice versa. It has happened in many places in the United States, around the Air Force or, you know, Army bases and everywhere. And it's not only in the United States, even in Germany, Japan, China, they have found, wherever you find TC or DCA, you will find 1.4 dioxane. So you expect to have it also in the rivers, in fish, and in the drinking water. Here is the map from the UCMR3, and you can see there are white dots, gray dots, and red dots. And essentially, this is above, below, and or around the recommendation concentration, which is 0.36 micrograms per liter. This is a reference dose this is the dose that it can cause 1 cancer per million, okay? And again, this is advisory, nothing regulatory. The areas we have chosen, as it was here, you cannot believe. If you live in Long Island, public water from Long Island comes from well water. And most of these areas, they're really high levels of 1.4 dioxane. And what happened on that? Actually, Governor Cuomo was very strong and put a legislation. And there is a law in New York now that prohibits the manufacturers to put 1.4 dioxane in detergents and household items. Now there is a lawsuit from the industry against the government, but you know, at least they put that, and they recognize that, because everything, and I don't have time to go through that, your Tide, your shampoo, your everything, it has 1.4 dioxane, and everything is going to go down to the water table, and especially in Long Island, the public water comes from well water. So it's a major concern. Another area which was one of the reasons that NIH had a very strong also thing is NIHS locates in North Carolina. Major contamination also in North Carolina, in the Cape Fear River is fully contaminated, and you know, it's still getting a lot of discharges in there. Is it only there? No. We have the case of Michigan, which I'll explain to you later. We're working on that. Ann Arbor has a major plan of 1.4 dioxin sitting right there, and there's a lot of concern. New Jersey found out last couple of years that the public water had 1.4 dioxin. And what happened? There is a lawsuit. I haven't followed up the details, but in March 23 of 2023, the water companies sued the manufacturers for putting 1.4 dioxin into the river, which eventually ended up into the drinking water. So there's a lot of things going on, and this is happening just right now. That was again in a different area in North Carolina, where they found again 1.4 dioxin, 1,300 times higher compared to the reference level. So what I'm saying is there is a lot of issues in there simply because nobody was paying attention before, and this is something that due to the difficulties in determining that and having the assays, but eventually right now there is a major concern. So in terms of toxicity, in general, most of the toxicity, what we know or what we knew and what we're going to do is from liver and kidney. Okay, but however, they have been found that there is some effects also in nasal and eye. Liver toxicity is dose-dependent, characterized by cell degeneration, pre-neoplastic lesion development, lobular swelling, necrosis, increased DNA synthesis, all the pre-neoplastic damages that you can see in chromosomal damage and enzyme leakage. The kidney toxicity manifests as a degeneration of the cortical tube cells, tubular necrosis, and glomerulonephritis. So the other thing that we have discovered in our, and which I think it could be very disturbing, but is that we found out that there is a potential disruption of glucose homeostasis, at least in our mice. But again, it's not published. We have it here. So the studies, and I'm not going to go, I can bypass because time is running. There are a lot of experiments, both in mice and rats in over the 2 years, that it is a proven liver carcinogen. And, it has been shown that, it is occurring in various labs throughout the globe, and they found the same thing. So one of the things, though, that it was a little bit puzzling is when they did the genotoxicity, in other words, they did the Ames test or sister chromatin exchange test, they couldn't find any genotoxicity for this 1.4 dioxin. So they said, well, it's not a mutagen. Maybe it's a promoter, as you can see on the fourth bullet. Well, there is a study published last year by a Japanese group that indicates that 1.4 dioxin can induce DNA adducts as well. Okay, and they're going on that. But what really triggered my interest, and this is what really reflects essentially the mode of action of 1.4 dioxin, is what they did is they took genotoxic carcinogens, not genotoxic carcinogens, and 1.4 dioxin. And they did the studies in both mice and rats, and they measured the RNA-seq, and they tried to make sense if 1.4 dioxin belongs to one or the other group based on gene expression. And what they found, as you can see from the slides, 1.4 dioxin is a distinct form. So in other words, it does not belong to any of these two models, which I thought it was very interesting and it was worth of exploring. So let's go and see what we're doing. So our project one briefly goes on liver, cancer, and biomarkers. So essentially, we're trying to use mouse models, human cells, and organoids, and also zebrafish to dissect the molecular mechanisms of causing cancer. That's what the project one does. The project two, as I told you, there is no epidemiological study on 1.4 dioxin. And this was the major obstacle that I had to go through for the resubmission, because everybody, when I was putting the stuff about carcinogenicity, they said, well, we don't have epidemiological studies. Well, somebody has to do it. Anyway, to make a long story short, the budget of this project is not huge, so we did not have money to do the epidemiological studies. However, the NIH said, we do want you to do something about it. So what we decided was exposure assessment. So in other words, epidemiological studies in much less number of samples. So the project 3 is something that we're trying to detect the 1.4 dioxane and co-contaminants in areas that they're there. My whole idea in here is, can we develop a system that it can be online, that you can monitor the area, do the biomonitoring from your computer, or the EPA can, get the information. It's a difficult task. Many eliminated Jordan Peccio, when I talked to them about it, they said, it cannot be done. Anyway, to make a long story short, I'll show you what we're doing, and we're trying to develop that. But the idea of having those sensors is really hot right now in all the aspects that you can do. Finally, the project 4, as I told you, and this is another very important project, is how we can degrade. So your well water most likely might have 1.4 dioxane. I'm not saying it does, but if it does, how you can purify it? Well, you can purify it with reverse osmosis, but do you have $15,000 to put just for reverse osmosis on your water? And some people may afford it, but how about the people that they cannot? We're talking about environmental justice as well. So there's a lot of issues that need to be discussed in there. So that's project 4. And the project 4, essentially what it tries to do is trying to use what we call advanced oxidation, which is essentially oxidative stress. And I have more slides to show you, but I want to give you the big picture. So you try to utilize hydrogen peroxide, you break the hydrogen peroxide, you create reactive oxygen species, and then the reactive oxygen species, they can degrade your chemical. And this is also, we're trying to do something similar with PFAS as well. But anyway, this is off the record. Let's go for project 1. This is the team that we have. Essentially, Yin Chen leads that. I'm a co-leader, but I'm following her. That's her project. And we have, of course, Georgia from and other people in the lab. And this is in collaboration with the National Toxicology Program, with Stephen Ferguson, who is doing the human hepatocytes and also the 3D structures of the human cells, that we can do the organoids and we can test 1.4 dioxane in there. So what we do here at Yale, and I'll show you, is we're doing the mouse work. So NTP, National Toxicology Program, is helping us with determining the effects on cells. And Robin Tangway from Ohio State University, which I was there 10 days ago, they're evaluating our 1.4 dioxane, but most importantly, the mixtures with the other co-contaminants on zebrafish. Why we do that? Because to do that in mice, you need, about 20-fold budget and much more time to evaluate that in mice. So zebrafish is an amazing tool, and actually her facility up in Oregon State for zebrafish screening, and especially the exposomic studies, is truly amazing. So we take advantage of that, and actually we're not restricting the science only to carcinogenicity, but we're also doing a lot of stuff for behavioral stuff, and also for that could have an effect on mental issues of this compound. So what we really know, again, this is, I'm going to pass, I have covered that quite a bit. We know that causes cancer. We don't know the mechanism. 1.4 dioxane is a carcinogen in mice. We don't know what is doing and what is going on. So what we're doing? So we're doing animal studies. We're not just repeating what it has been done before, but we're trying to use knockout animal models that will have an effect on this. So we're using models on oxidative stress, and I will give you the examples in here. But essentially, the first part is we're using mice, and we do high dose and low dose. The high dose is to get the effect. Low dose is to mimic the human exposures. And from there, we're doing the omics-based system approach. We're doing the RNA transcriptome, the metabolomics, and also the phenotyping of these mice. And we have quite a bit of data so far. So then we combine this on using deep learning, doing the RNA-seq, the metabolomics, and also the the phenotyping things. And we're trying to determine the mode of action, the MOA, and also the exposure and toxicity biomarkers, which we're going to feed the project to when they do the exposure analysis. I told you before that we also using the HEPA RG cells with NTP and the zebrafish for the AIM3. Here is a little bit of what we know and what we're currently know regarding the metabolism of 1,4-dioxane. One of the things I want to have your attention to it. It is metabolized by cytochrome P452E1. Why this is important? Because cytochrome P452E1 is the activator of many carcinogens, many what we call precarcinogens, to very active intermediates that they can cause cancer, that can interact with DNA. So as you can see on the right-hand side, you can see also you have the TCE and the PCE, which are the solvents, which can also be metabolized by cytochrome P452. I want you to keep that in mind, because what we have found, it's something that I think we have explained some of these effects. So we've already published quite a few papers in terms of the mechanism. I wish I had too much time to show you, but one of the major findings that we did, and nobody has shown that before, is we found that 1,4-dioxane induces cytochrome P452E1. And why this is important? Because if you have a co-exposure of cytochrome P452E1 and trichloroethylene or diethylnitrosamine in your cigarette smoke or in the smoked food that you eat, then you have higher chances of metabolizing the procarcinogens to carcinogens, and they can cause liver cancer. So in other words, it can act as a promoter by inducing the cytochrome P452E1. In addition to the cytochrome P452E1, you can see it in your left, we find that there is increased oxidative stress as indicated with 4-hydroxynonenal, and also with the increase of the quinone oxidoreductase, which is a gene involved in the antioxidant response. So I apologize, this happens again. The image becomes a little bit, but what the highlights of our research are this, and this happens to me. I don't know why, but I have another one that I can show you. But we have found out so far that there is a direct genotoxic effect for dioxin that includes oxidative stress. We already published that in 2022. There is a dominant role of 2E1 in the metabolism, as we have found by metabolomics, and also for the liver toxicity. But also we have not only the induction of 2E1, we have found a second mechanism which is completely independent of cytochrome P452E1. How do we do that? We're using knockouts that they have no cytochrome P451, and we determine that. So this is what we're doing. Specific AIM1 and specific AIM2 is we're using knockout mice. And again, remember, we have the metabolism here. As you can see, cytochrome P450 is metabolized. The first two animal models, the GCLM knockout, is a model of mice that has low glutathione levels. So that animal model, it has low antioxidant capacity. NERV2 knockout model is the NERV2 is a transcription factor involved in all the genes involved in the antioxidant response. So if the NERV2 gets activated, your cell becomes really active against the insults. And we're currently using that. We're doing the experiments and to do that. So we're also using the cytochrome P452E1. As I said, we completed those studies actually. And we're using the NQ1 and also the aldehyde dehydrogenase 2. As you can see, the ALDH2 could be involved in the last steps of the metabolism of that. And, the ALDH2 is a gene that it's highly polymorphic in a lot of individuals, especially those from Asian population, quite a bit. So project 2 is the exposure assessment team, co-led by Nicole Dizer and Brian Lederer. And we have also most of our people from our department in exposures, Crystal Pollitt, Zay and Lou, and also we have collaborators in North Carolina State, John Hoppin and Detlef Napi. And this, as I told you, is going to be an exposure model, exposure assessment model here in Long Island, and we have already going through that. So the aims again is you go, you collect the water, you collect the blood from these people, and then you're trying to also match the medical records, and you're trying to do the exposure assessment. And we're trying to interact all this project, as I told you, is we're using metabolomics in this project, and the metabolomics here will be coordinated with the metabolomics of the first program, which is on the first specific aim, which is in mice. The third project is led by Manny Elimelech from the School of the Department of Chemical and Environmental Engineering, and also, is Jordan Pechia and True Gender are from this group. And again, what they're trying to do is they're trying to develop this kind of molecules, the heptamers, or all this idea that it can bind to that, and then it can be detected, and then they can transmit that. It's tough. We're not going to accomplish in the first 5 years, but at least we can develop the sensors to develop, and then we can try to find out how we can develop the network. Fourth project, and I apologize, I'm trying to finish so we can have some time for discussion. Fourth project is by Jae-Hong Kim and John Fortner. And as I told you, this is relating to develop small devices that you can use in your house to get rid of 1.4 dioxin. And they're using the advanced oxidation. It will take me about a lecture to explain you that, but essentially, I think I told you the principle is hydrogen peroxide generate hydroxy radical, and this hydroxy radical will hit that. They're using actually two kinds of approaches in there. And I think I have, they are developing some nice things, and I'm gonna
Let me share some recent progress that we have made. We successfully synthesized the catalyst, boron-doped carbon catalyst shown here, and fabricated a hydrogen peroxide synthesis cell. We quickly discovered that maintaining the performance in real water metrics would be the key for success of this research, to provide a system that can perform for a long period for household application. We therefore developed a framework on how to optimize pulsed electrolysis to enhance catalytic tolerance against impurities present in water and to improve overall lifetime of the cell. We discovered that optimized pulsing sequence enabled improved long-term hydrogen peroxide performance to nearly 300 hours and 35 times better than conventional electrolysis, even in the presence of most detrimental impurities such as nickel and zinc. These findings make this cell closer to real-world implementation for prolonged hydrogen peroxide synthesis and subsequent 1,4-dioxane destruction. Let me share some recent progress that we have made. We successfully synthesized the catalyst.
I'm sorry. It was. I thought it was.
As an alternative.
This is the second method
to destroy 1,4-dioxane. In specific aim 2, we explore the use of engineered gas phase nanobubbles. We performed extensive characterization of nanobubbles in solution with a focus on hydroxyl radical generation, and by performing a number of different characterization using degradation of hydroxyl radical specific target compounds, electron paramagnetic resonance spectroscopy, and a fluorescence-based indicator. Through this phase of research, we concluded that nanobubble-induced hydroxyl radical generation is minimal, if not all, but we will continue to study if there is an alternative way to enhance the nanobubble-enabled advanced oxidation.
So
As an alternative technology.
I wouldn't be able to tell you that. So that's why I sent my daughter. My daughter is first year of environmental engineering in Boulder, Colorado. So the next is our community engagement core, which we have Iris Kaminsky and Andrea Esposito, co-leader, which executive director of the Citizens Campaign of the Environment. And she's very big in terms of community engagement. And we're doing quite a bit in community engagement. We're ready, and we utilize community engagement actually to recruit people for the project too. This has been fascinating so far, but we're not staying only in Long Island or even Vermont. We're expanding in other areas too. So the other areas that I'm working right now, and I will tell you, is the New Hampshire, in North Carolina, Michigan, and recently I have been engaged by Florida, believe it or not. There is an area over there, Lake Mary, that has even 30,000 times higher levels of 1.4 dioxin in their surface water. Anyway, this is some of the stuff that we already done and the publicity that the center is getting, and I have more and more, but I'm just leaving you that. We're using that as a tool, and we communicate. People calling me in the office, they said, we want to register for this study, and they need more information. And this has not been studied only in Long Island. And, newspapers have taken the center because of the importance, as I told you, of 1.4 dioxin discovered in many states. So we're getting a lot. Training component, I think I was smart on that. I took our I took Chris Chudy. Chris Chudy is our esteemed director of a graduate program at YSPH, and also we have the engineer, a graduate program, and these two made the dream team. And it had actually one of the best scores on the components. They had almost 10, 12 on their application, and Yin Chen is also co-investigator on that. So we building up a training educational capacity, going from even undergraduates with an R25 that I have on training other graduate students, to MPH students and also PhD students and postdoctoral. This is classic regular, the schedule we're doing for the training of our PhD. Last but not least, and I kept the picture in here of Peter Petuzzi, although he retired and he just stepped down right now. But I love Peter, and that was another dream team here of Hong Yu Zhao and Peter Petuzzi, that they do the data management and analysis core team, which essentially bring all the projects together. And I'm not going to go to all specific aims, but essentially is coordination between projects and cores, fostering data sharing and interoperability. So we're trying to develop all these cloud systems. And finally, and most importantly, is data quality assurance, quality control, and data integration. So this is huge, and we get really good. So I am almost at my 45-minute mark. And essentially what I would like to say, the establishment of our Yale Superfund Center marks a significant milestone of our departmental commitment to addressing emerging contaminants linked to cancer. Our strategic plan includes the development of peripheral research project. We got a budget cut of almost 50% of our initial budget. Not only us, everybody did, because they withdrew some money from that project to support the climate change in a number of institutes. So what we're trying to do is we're trying to develop peripheral research projects for exposure assessment on various locations across the United States, fostering international collaboration and broadening the impact of our work. We aim to strengthen the partnerships, of course, with Cancer Center, with the Liver Center, with Diabetes Center, leveraging their expertise and resources for more comprehensive approach to our research and vendors. And as we advance, our focus will extend beyond the scope of the 1.4 dioxin and the volatile compounds. And we're going to try to get more emerging contaminant with particular emphasis to PFAS. And actually, PFAS is, because of their ability to be endocrine disruptors, they have been linked now to obesity and diabetes. And as I told you before, we find something similar. And there are a lot of interactions. Another thing about PFAS induces kidney cancer, okay? It's the major cancer that induces is kidney cancer. So what is the interaction of this between them? This is something that we need to explore. So this expansion will involve in-depth exploration between links and the PFAS, especially, as I told you, kidney cancer and obesity, bringing valuable insight to the scientific community. I'm going to stop with that, and I'm going to take questions, and we're happy to discuss anything you wish. Yes.
Great, thank you. I appreciate the whole project began with the involvement of the community alerting you to 1,4-dioxane. So at the Cancer Center, liver cancer is a priority cancer that is in the strategic plan, given its increased rates in Connecticut as well as nationally. So I'd love to think about collaborations to how we can do more on the epidemiologic or clinical aspect and
Absolutely.
link it. With patients coming in newly diagnosed with liver cancer, could there be a case control study where water is collected from their home, blood, questionnaires, and then have a control sample? So there's so much opportunity, I think, here.
That's what David said. That's why he invited me to give the talk here. We need to get this. Melinda, this is a very good point. If we can get more and actually, we can explore the possibilities, and if we have more blood samples of that, we can do much better. Much more. And that's a very good point. Yes.
Going to Long Island for Thanksgiving.
Get your water with you.
Is there a safe, what do you recommend? Is there a brand of bottled water that's guaranteed to not be contaminated?
It depends now. Listen, I mean, there are areas in Long Island. There is this interactive map that you can find which areas they have high levels. But to be on the safe side, I was going to say use public water, but this public water over there comes from well water. I don't know how the late status is, but it might not be a bad idea to use some bottled water. David.
First of all, I want to echo what you and Melinda said, because there are tremendous opportunities here that are aligned in many ways with the forthcoming Cancer Center strategic plan. I'd like to add to that education, because you have cancer education programs running that are cancer-connected education programs running that are complementary to our other training programs. The question for you, though, is you began with a challenge from the state of Connecticut, but I noticed all the collaborations are out of state. Can you tell us more about how this work might impact our state and our catchment community, which is the same thing?
There are certain areas that they have 1,4-dioxane in the state of Connecticut. However, the state of Connecticut has taken very good care of their public water, and their public water is pretty safe. As long as you are on public water, they're taking good care. How it's going to have a major impact? It's going to have a major impact on liver cancer. But, the problem, it might not be completely here, but it has several aspects of, getting engaged with agencies, the state agencies addressing that. And as Melinda said, there is an increased rate of liver cancer, which brings the other point you were talking about. I also have a T32 program with livers, with psychiatry essentially. But it's my point of view is my other lab that I have is alcohol and cancer. And I actually organized the International Conference on Alcohol and Cancer. So this is another area that I think the increased levels of alcohol consumption is a very good contributor in that, along with obesity. So if you add another factor, which is 1,4-dioxane, even if it's in low levels, for example, in the state of Connecticut, what we saw in here, there is an increase of the cytochrome P452 So if you get that in combination with smoking, nitrosamines, or other exposures, you can increase the rate of, of cancer in the area. Right? Yes. Well, that's a good point. Listen, we have done a lot of risk evaluation risk for water sources on Welsh water. I wouldn't say that 1.4 dioxin is that major concern because we know what were the areas of 1.4 dioxin. My concern would have been more on the PFAS. So my recommendation is test your well water for PFAS. That's the only suggestion. I don't think in Connecticut we have that major issue of 1.4 dioxin. Well, if you test, that's a very good point. If you test your well and it's positive for 1.4 dioxin, you don't have to wait for Jae-Hong Kim to develop these devices. What you do is you use plastic water. I mean, plastic water from from bottles. Of course, you can have some things from there, but it's at least safer. If you find that there is PFAS, the PFAS you can filter them with charcoal and stuff like that. So there are devices that are relatively cheap. But one of the things that I want to tell you is the importance of drinking water, because you're going to drink, you may avoid drinking alcohol, you may avoid smoking cigarettes, but you're going to drink your water. Yes. You're safe. You're safe. You cover both of them. Well, the other thing is, in terms of the cancer center, is the liver center, and also there is a high incidence of alcohol-induced liver cancers. And not only the liver cancers, but also colorectal cancers. And the incidence of colorectal cancers are really high as well throughout not only the nation, internationally, and especially the early onsets. Yes. I think so. Yes. So thank you for the question. Usually the stages are, you start from steatosis, you go to fibrosis, you go to cirrhosis, and then in some cases, you go to hepatocellular carcinoma. It is plausible that you can go without cirrhosis, yes. I mean, in animal models, they go without any signs of cirrhosis at all. But one of the problems, well, talking about animals and humans and stuff like that, that alcohol, it is well known that causes liver cirrhosis, right? If you try to do the same thing in mice, there is no way you can do it. But you can take a mouse and you can put carbon tetrachloride for 3-4 weeks, you'll get cirrhosis 100%. So that's the challenge that you have between animal models and human thing, and that's what we're trying to do the organoids as a complementary to the mouse studies. All right, thank you very much.